This lesson examines the mechanisms by which substances are transported across the plasma membrane. We'll explore the different types of transport mechanism and how each transport mechanism moves materials, focusing on differences in transported materials, concentration gradients, and energy requirements. Cell membranes are selectively permeable, meaning they allow certain substances to pass while blocking others. The arrangement of the phospholipids and proteins that make up the membrane structure enable the membrane to carefully regulate the movement of materials into and out of the cell. The phospholipid bilayer allows small, nonpolar molecules such as oxygen, carbon dioxide, and small hydrocarbons like methane to pass through the membrane easily. This is because the size and lack of charge of these molecules let them dissolve in the membrane's hydrophobic interior and pass through the membrane by simple diffusion. At the same time, the hydrophobic interior of the bilayer acts as a barrier to ions and larger polar molecules, preventing these substances from passing freely. It should be noted that small amounts of water can still pass slowly through the membrane by simple diffusion. Some transmembrane proteins, like those shown here, act as transport proteins that allow these hydrophilic molecules to move across the membrane's hydrophobic interior. Aquaporins, for instance, are specialized transport proteins that enable water molecules to pass through the membrane at a faster rate than the molecules would move by simple diffusion alone. There are two major types of transport proteins: channel proteins and carrier proteins. As you can see, channel proteins are embedded in the membrane and contain a tunnel lined with charged and/or polar hydrophilic amino acids. Hydrophobic regions of the protein hold the protein stably within the lipid bilayer, while the hydrophilic tunnel, lined with charged and/or polar amino acids, allows hydrophilic substances such as ions and polar molecules to pass through the membrane. Carrier proteins are also embedded in the membrane, but instead of providing a hydrophilic tunnel, carrier proteins transport specific solutes by first binding to them. This binding triggers a conformational change in the protein, causing it to open on the opposite side of the membrane and release the solutes. Once the solutes are released, the protein returns to its original shape, ready to bind to another solute specific to that carrier. Channel and carrier proteins are usually specific for the substance they are meant to transport. In fact, most transport proteins are specialists. Each one is designed to move only one particular substance. Some transport proteins handle ions like sodium or potassium, while other proteins carry small molecules, such as glucose. Whether a cell transports a certain substance into or out of the cell typically depends on whether the specific transport protein is present in its membrane. Transport across membranes may be passive or active. Passive transport occurs spontaneously as solute molecules move down their concentration gradient from an area of higher solute concentration to an area of lower solute concentration without energy input from the cell. Think about riding a bike. When you're going down a hill from the higher elevation to a lower elevation, you don't need to expend energy pedaling. Gravity does the work for you. But what about when you have to bike uphill? In that case, you have to expend energy to move the pedals to get from the bottom of the hill to the top, moving from a lower elevation to a higher elevation. This is similar to active transport, where the cell moves molecules against their concentration gradient from an area of lower solute concentration to an area of higher solute concentration, which requires an input of cellular energy, typically in the form of ATP. Now that we've got the basics nailed down, let's examine cellular transport in more depth. Simple diffusion is one mechanism of passive transport used by cells. In simple diffusion, small, nonpolar molecules, such as oxygen and carbon dioxide, move down their concentration gradients directly through the phospholipid bilayer without the need for transport proteins or cellular energy. Facilitated diffusion is another type of passive transport and is used to move ions and small polar molecules through the cell membrane. These hydrophilic solutes cannot pass through the hydrophobic core of the phospholipid bilayer and therefore require a transport protein to help cross the membrane. Movement of solutes in passive transport is driven by concentration differences. So, ions and small nonpolar molecules pass through the transport protein down their concentration gradients without the need for cellular energy. Although the passive transport of water molecules through aquaporins represents a type of facilitated diffusion, water is so essential to life, and its movement into and out of the cells is so consequential that it is often considered separately from the transport of other molecules. The passive movement of water molecules across a selectively permeable membrane, either by simple diffusion directly through the phospholipid bilayer or via facilitated diffusion through transport proteins called aquaporins, is known as osmosis. Even though osmosis can occur directly through the phospholipids without the use of transport proteins, our focus is on movement through aquaporins because they provide a faster and more efficient pathway for the water transport across membranes. Although osmosis is the movement of water from a higher water concentration to a lower water concentration, in cells, the net direction of water movement is often easier to predict by comparing solute concentrations on each side of the membrane. Let's look at why the presence of solutes matters for water movement. When solutes are present, some water molecules form hydrogen bonds with the solute molecules. Aquaporins are narrow protein channels that allow only individual free water molecules to pass through in single file. So, water molecules bound to solute particles are too large to enter the aquaporin and cannot cross the membrane. As a result, a higher solute concentration means fewer free water molecules are available to move across the membrane. On the other hand, since the other side of the membrane has a lower solute concentration, there will be more free water molecules available. As a result, free water moves down its own concentration gradient from the side of the membrane with a lower solute concentration and higher free water concentration to the side of the membrane with a higher solute concentration and lower free water concentration. This movement of water molecules continues until a dynamic equilibrium is reached, meaning the concentration of free water molecules is the same on both sides of the membrane. Let's look at a different diagram to see what this might look like. Here we see a beaker of water with a semi-permeable membrane that allows water molecules to pass through, but blocks the passage of solutes. When one side of the membrane has fewer solutes and the other side has more solutes, water flows from the side with a lower solute concentration, where there's higher free water concentration, to the side with a higher solute concentration, where there's a lower free water concentration. In other words, water moves down its own concentration gradient through the process of osmosis. As osmosis occurs, the water level on the side of the beaker with more solutes will rise. This continues until the concentration of free water molecules is equal on both sides of the membrane, a state known as dynamic equilibrium. At this point, water molecules continue to move across the membrane in both directions, but there is no net movement of water. The same principles of osmosis that we observed in the beaker also apply to living cells. In cells, water movement depends on the relative solute concentrations inside the cell compared to the relative solute concentrations in the extracellular fluid surrounding the cell. The ability of the extracellular solution to cause the cell to gain or lose water is called tonicity. Tonicity depends on whether solutes in the extracellular fluid can cross the cell membrane. Only solutes that cannot cross the membrane influence water movement. Let's look at tonicity and how it can affect cells, particularly animal cells. In practice, tonicity of a solution is usually described in one of three ways: isotonic, hypotonic, and hypertonic. We'll explore the effects of these types of solutions on animal cells using a red blood cell as a representative example. An isotonic solution is one in which the solution outside of the cell, such as blood plasma in our example, has the same solute concentration as the solute concentration inside of the cell, that is, the red blood cell cytosol. In Greek, iso means equal, so the same solute concentrations inside and outside the cell makes sense. When a cell is in an isotonic solution, water enters and leaves the cell at the same rate, so there is no net change in the cell's volumes. Hypotonic solutions affect cells differently. Hypo means below in Greek, and hypotonic solutions have a lower solute concentration than the solute concentration inside the cell. This difference in concentration causes a net flow of water into the cell, which causes the cell to swell and potentially burst, a process called cell lysis. In organisms whose cells have cell walls, such as bacteria, plants, fungi, some protists, a hypotonic environment, such as the one shown surrounding this plant cell, typically does not cause the cell to lyse because the cell wall is rigid and limits cell expansion, which limits water intake. Water entering the cell pushes against the cell wall, creating turgor pressure, which helps keep the cell firm and opposes continued water uptake by the cell. So what happens if cells are placed in the third type of solution, a hypertonic solution? Hyper is Greek for above, and in this case, the solution outside of the cell has a higher solute concentration than the solute concentration inside the cell. This difference in concentration causes a net flow of water out of the cell and into the surrounding solution. As a result, the cell may shrink and become crenated. The tendency of a solution to cause water to move through a semipermeable membrane, such as the plasma membrane, is called osmotic pressure. Let's try to visualize osmotic pressure using a diagram. Here, we see a beaker divided by a semipermeable membrane that allows water molecules to pass through, but not solute molecules. The left side of the beaker contains a hypotonic solution with a lower solute concentration than the right side of the beaker, which contains a hypertonic solution with a higher solute concentration. The difference in solute concentrations across the membrane creates osmotic pressure, causing water to move across the membrane from the hypotonic side to the hypertonic side by osmosis. Osmotic pressure can lead to significant shifts in water volume within the cell, so regulating water balance is essential, especially for animal cells which lack the protection of a cell wall. Different organisms use various osmoregulatory strategies to keep water and solute levels in balance. For example, in humans, the kidneys help regulate body water by controlling how much water is reabsorbed from the filtrate back into the blood, and thus how much water is ultimately excreted from the body. We'll cover kidney function in more detail in another lesson. Now that we've explored the different types of passive transport, let's turn our attention to active transport and take a closer look at how it works. As mentioned earlier, active transport happens when a cell uses energy to move substances against their concentration gradients, from an area of lower solute concentration to an area of higher solute concentration. Cells can move small solutes or ions in this way using transport proteins, similar to those used in facilitated diffusion, but in active transport, these proteins require energy to work. So what forms can this energy take? In primary active transport, the energy to move substances against their gradients comes directly from the hydrolysis of ATP, which is why it's called primary active transport. Hydrolysis means splitting a molecule using water, in this case breaking ATP into ADP and inorganic phosphate. Hydrolysis of ATP releases energy when the bond between the second and third phosphates in ATP is broken. In primary active transport, part of the energy from the hydrolysis of ATP is used to attach the phosphate group that was freed from ATP to a transport protein. This phosphorylation step changes the protein's shape, causing the solute to cross the membrane. In some eukaryotic cells, especially in animals, an important primary active transport system is the sodium-potassium pump. Let's see how this transport system works to actively transport sodium and potassium ions. The sodium-potassium pump is a membrane transport protein that alternates between two main conformations. In its initial state, shown here, the pump is open towards the cytosol of the cell. In this orientation, the pump's binding sites have a high affinity for sodium, allowing 3 sodium ions from the cytosol to bind. The binding of 3 sodium ions stimulates the hydrolysis of ATP, which transfers the phosphate group to the pump. Phosphorylation shifts the pump's conformation to face the extracellular space, which decreases the pump's affinity for sodium and promotes the release of the 3 sodium ions into the extracellular fluid. In this outward-facing conformation, the pump has a high affinity for potassium, allowing 2 potassium ions from the extracellular fluid to bind to their specific binding sites on the protein. The binding of potassium triggers the release of the phosphate group from the pump. Dephosphorylation causes the pump to revert to its inward-facing conformation, which reduces the pump's affinity for potassium and causes the release of the 2 potassium ions into the cytosol. In this conformation, the pump again has an increased affinity for sodium, and the cycle can begin anew as long as ATP is readily available. For each ATP that is hydrolyzed, the sodium-potassium pump moves 3 sodium ions out of the cell and moves 2 potassium ions into the cell. The sodium-potassium pump moves sodium and potassium against each ion's concentration gradient, maintaining a high extracellular concentration of sodium and a high intracellular concentration of potassium. These gradients are vital for many cellular functions, including generating the electrical signals used in nerve impulse transmission. The sodium-potassium pump is not only an example of primary active transport powered directly by ATP, but also provides an example of an antiport. An antiport is a membrane transporter that moves 2 or more solutes in opposite directions. For example, the sodium-potassium pump moves sodium out of the cell and potassium into the cell. In contrast, a uniport moves only one type of solute in a single direction. For instance, the plasma membrane calcium ATPase uses ATP to pump calcium against its concentration gradient out of the cytosol. Lastly, symports move 2 or more solutes in the same direction across the membrane, as demonstrated by the sodium-glucose link transporter, also known as SGLT, which carries both sodium and glucose into the cell together. We'll discuss the sodium-glucose link transporter in more detail shortly. It should be noted that even though we are discussing these transport systems in the context of active transport, uniports, symports, and antiports can also function in passive transport. So far, we've focused on primary active transport, where ATP directly powers the movement of molecules across the membrane. Now let's take a look at secondary active transport. Secondary active transport is a form of coupled transport in which one solute moves by passive transport down its concentration gradient, releasing potential energy that is harnessed to actively transport another solute against its gradient. With these key ideas in mind, let's explore an example that shows how secondary active transport works in cells. As noted in our overview of symports, cells use the sodium-glucose link transporter, also known as SGLT, to move sodium and glucose into the cell together, an excellent example of secondary active transport. Let's look more closely at how SGLT carries out secondary active transport. As described earlier, the sodium-potassium pump represents a primary active transport mechanism that uses the energy released from ATP hydrolysis to maintain a high extracellular sodium concentration. When sodium ions diffuse back into the cell down their concentration gradient, the potential energy that is stored in the sodium concentration gradient is harnessed by the SGLT transporter to move glucose into the cell against the glucose concentration gradient. This movement of glucose is considered secondary active transport because the energy driving glucose uptake comes indirectly or secondarily from ATP hydrolysis through the sodium gradient established by the sodium-potassium pump. As you can see, secondary active transport is a useful strategy that allows cell to borrow the energy stored in ion gradients without directly using additional ATP. We've seen how concentration gradients can power transport, but ions also carry charge. This means their movement doesn't just create chemical concentration differences, it also generates electrical gradients across the membrane. Let's see what this means for cells. When ions, such as sodium and chloride, are transported across a membrane, they can create an unequal distribution of charge between the inside and outside of the cell. This difference in charge is called an electrical gradient. For example, if the inside of the cell is more negative than the outside, as is shown in this image, positively charged ions, such as sodium, will tend to move passively into the cell, while negatively charged ions like chloride will tend to move passively out of the cell due to being attracted to the region of opposite charge on the other side of the membrane. Because sodium and chloride are charged, this movement typically happens through passive membrane transport proteins known as leak channels. Here we see the passive movement of sodium into the cell and potassium out of the cell. The image is simplified, since in reality these ions travel through channel proteins rather than moving directly through the phospholipid bilayer. The passive movement of ions like sodium and potassium across a membrane is influenced by two factors. The first factor is the ion's concentration, or chemical gradient, which reflects the difference between high and low concentrations of ions on opposite sides of the membrane due to the sodium-potassium pump we discussed earlier. The second factor is the electrical gradient, which results from the charge difference across the membrane. Because sodium and potassium are positively charged, both are attracted to the negatively charged interior of the cell. Combined, the chemical and electrical gradients make up what is referred to as the electrochemical gradient. An important point to remember is that the electrical gradient, such as that of potassium in this image, can sometimes work against the chemical gradient, which can affect the speed of transport or even reverse the direction of transport. For instance, in this cell, the concentration of potassium is higher in the cytosol than in the extracellular space. This concentration gradient would tend to cause potassium ions to passively move out of the cytosol and into the extracellular fluid. At the same time, the inside of the cell carries a net negative charge, while the outside has a net positive charge. It should be noted that the cytosol remains negative overall despite its high potassium concentration due to the presence of large negatively charged molecules such as proteins that cannot cross the membrane. The difference in electrical charge across the membrane constitutes an electrical gradient that tends to drive the positively charged potassium ions back into the cell. The combined effect of these two forces, that is, the electrochemical gradient, still favors the movement of potassium out of the cell, but less strongly than the concentration gradient alone would suggest. A key factor in maintaining the electrochemical gradient is the operation of the sodium-potassium pump, which, as we discussed earlier, moves sodium out of the cell and potassium into the cell. This activity maintains a high concentration of positive ions outside the cell and leaves the inside relatively negative, creating a voltage difference across the membrane known as the membrane potential. The membrane potential is vital for cellular function as it drives processes such as secondary active transport of glucose through SGLT into intestinal epithelial cells after consuming a meal, and the propagation of nerve impulses along neurons. So far, we've focused on how cells move individual ions and small molecules across membranes. But cells also need to transport much larger molecules in and out, which requires a very different strategy called bulk transport. Large biological macromolecules, such as low-density lipoproteins, or LDLs, are not typically moved across the plasma membrane by membrane transporter proteins. Instead, these substances are taken up through bulk transport, which means the cell internalizes material in vesicles, either capturing many molecules at once, as with LDL, or engulfing large particles such as bacteria. The bulk transport of substances into cells in small vesicles is called endocytosis. During endocytosis, a particle or macromolecule to be transported becomes enclosed in a pocket formed by an extension of the plasma membrane. In an energy-dependent step, often requiring ATP or GTP, the sides of this pocket pinch together and seal off, creating a membrane-bound vesicle known as an endosome inside the cell. The contents of the endosome are then delivered to the appropriate location through the endocytic pathway, which is discussed in more detail in another lesson. Endocytosis can occur in several forms, depending on both the size of the material being internalized and whether the process is specific or nonspecific. Endocytosis of extracellular fluid containing small solutes is known as pinocytosis, or cell drinking. During pinocytosis, the cell engulfs extracellular fluid along with the solutes dissolved in the fluid. In pinocytosis, solutes do not need to bind to specific receptors for an endosome to form, which makes pinocytosis a nonspecific process. In contrast, the uptake of very large particles, such as microorganisms or cellular debris, is called phagocytosis, or cell eating. Phagocytosis of large particles is generally a specific process, as it usually requires receptor binding, followed by extensions of the plasma membrane called pseudopodia that surround the particle. The enclosed particle forms a vesicle called a phagosome, which is then drawn into the cell. Specialized phagocytic cells, including macrophages and neutrophils, play a critical role in innate immunity, which is discussed in more detail in another lesson. One type of endocytosis that targets specific molecules through receptor binding is known as Clathrin-mediated endocytosis. Clathrin-mediated endocytosis is a highly specific process in which cell surface receptors cluster in Clathrin-coated pits, which are specialized regions on the cytosolic surface of the plasma membrane that are lined with the protein Clathrin. A key example of Clathrin-mediated endocytosis is the uptake of cholesterol from the bloodstream. Since blood is mostly water and cholesterol, as a lipid, is insoluble in water, cholesterol molecules must be transported within particles called low-density lipoproteins, or LDLs, which we discussed earlier. LDL particles consist of a phospholipid monolayer with apolipoprotein B, referred to as ApoB, incorporated into the monolayer. During Clathrin-mediated endocytosis, ApoB binds to LDL receptors located in Clathrin-coated pits, triggering formation of a pocket. When the pocketed section pinches off, a vesicle that is fully coated with Clathrin is produced. This vesicle then delivers the LDL particle to the cell's interior where cholesterol can be released for use. Just as endocytosis allows cells to bring material in, cells also need a way to move substances out. This outward transport of molecules in vesicles is known as exocytosis, and it serves as the functional counterpart to endocytosis. In exocytosis, intracellular secretory vesicles carrying molecules destined for secretion are transported to the plasma membrane. The secretory vesicle then fuses with the plasma membrane in a process that requires ATP and specialized fusion proteins. Once fusion occurs, the vesicle's contents are discharged into the extracellular space, and the vesicle membrane, including any proteins or lipids it contains, becomes incorporated into the plasma membrane. Exocytosis is essential for the secretion of cellular products such as water-soluble hormones, including insulin and human growth hormone, digestive enzymes such as pepsin, and neurotransmitters like acetylcholine, which signal other cells. Exocytosis is also used in the elimination of cellular waste and toxic substances, and plays a role in remodeling the cell membrane. Specifically, exocytosis is used to add proteins, such as transport proteins and receptor molecules, to the plasma membrane, as well as bringing lipids and carbohydrates into the membrane. Although the details of bulk transport and the many other forms of membrane transport are varied, these mechanisms are critically important for the survival of cells and the function of entire organisms. The table shown here summarizes the major types of transport that we have discussed. Together, these mechanisms highlight the efficiency and versatility of the cell, allowing it to transport substances ranging from the smallest ions to the largest macromolecules.